Good morning, good afternoon, good evening, everybody. It's my pleasure to welcome everybody to another talk in Global IMMUNOTALKS 2023. My name's Laura McKay. I'm from the Doherty Institute at the University of Melbourne. And it's such an honor today on behalf of the 2023 Global IMMUNOTALK organizers to introduce our today's speaker, Professor Asya Rolls from the Technikon Institute in Israel. Now, Professor Rolls, she's well known for exploring the connection between the brain and the immune system, and we're so excited to hear about the groundbreaking science that we're going to hear about today. But just to give everybody a little bit of background, first to Asya's career. She was born in Russia, relocating to Israel, where she did her PhD at the Weizmann Institute under the mentorship of Michel Schwartz. And here she was really interested in studying how immunity impacts brain function, really getting on one of the very first people to get on the wave of neuroimmunology, which has really exploded in the past few years. From here, she went to do a postdoc at Stanford University, where she worked on the impact of sleep on immunity, and thereafter relocated back to Israel to start her own group at the Technikon Israel Institute, focusing from first how immunity impacts the brain, from how the brain impacts immunity. Now, she was one of the first to establish the link between the brain's reward system and immunity, or the placebo effect, and also she's had some groundbreaking work, which hopefully she'll tell us about, recently published in Cell, talking about immune memories in the brain, which we can't wait to hear more about. So rightly so, Asya is decorated with numerous awards. She was selected as one of the most influential scientists in Israel. She was also, she's a member of the Israel Young Academy of Science, as well as being a HHMI International Scholar, amongst many, many other awards and high impact publications. So welcome. We're so excited to hear about your science today. But of course, as a tradition here at Global IMMUNOTALKS, our listeners would love to get to know you just a little bit better and how you got to where you are today. And so the question that we've selected for you is, how would you describe that your personal style has impacted your career to date?
So first, thank you. I'm really excited to be here today. And it's a question that I think my personal style is in the sense that I really trust my intuition. And I think also a lot of the science that and the way we are doing science is based on this intuition, on this ability to sense that something is happening there, even if you still don't understand what's going on there, and to go with this gut feeling, which I think is something that it has to be a personality. So I think to some extent, I feel my postdoc advisor, Luis, who used to say that you feel comfortable out of your comfort zone. So I think in many ways, this is, if I can say, my style. I think this is also a style in science in many ways. It has limitations a lot, so I need to be very careful about that. But I think it's definitely a guiding direction.
Following your instinct, of course, for you to break the boundaries that you have and be such a trailblazer, you have to break the boundaries. And so following your gut instinct, that's such fantastic advice to really break the mold as you've done. And so hopefully people take that on board as great advice. So we're so looking forward to your talk. If you can share your slides,
okay,
that would be wonderful.
1 second. I thought anything. Perfect. Now you can see me well, right?
Perfect.
See the presentation? Right. Okay. So, okay. So thank you very much for this. I'm really happy to be here today and to talk to you about, I think, a concept that is kind of becoming more and more, develops more and more, is this immunoception. And I will get to it, I think, mostly in the end. But the overall idea that I hope you can see throughout the talk is that there is an ongoing dialogue between the brain and the immune system that is implemented in both systems. And there is a lot of similarities. When we talk about the immune system, we have this very classical view, right, of this initial innate response that is based on predetermined programs. Then we have this adaptive response, which is that the immune system kind of evolves and develops unique and more precise response to an antigen, but also it's the opportunity to cope with an antigen that the innate system never encountered before. So it can be like COVID in this case. And then this new learning process that was acquired during the adaptive immune response has to be learned, has to become a memory, which is something that it's very obvious for immunologists to talk about immune memory. But when neuroscientists talk about memory, they talk about something that in many ways is very much similar, but they're talking about the representation of experiences of different states and how it's encoded in the brain. But eventually, these are the two systems that have such a strong component of memory, the nervous system and the immune system. And they need it because they need to be able to cope with the unexpected. And this is exactly what I think this is the property, like the fact that we can cope with COVID and evolve and develop a very precise response to something that our ancestor never encountered. And the same way, when we are thinking now of landing on Mars, without knowing what's going to happen there, but we anticipate that, or we believe that our nervous system will be able to cope with these completely new environments and new situations and learn and adapt to this new environment. And these are very unique properties to both the nervous and the immune system. So I think it's not even surprising that they have to be in ongoing dialogue. And this is a bidirectional communication pathway. And of course, all this connection on how the immune system affects the brain, this was, as Laura mentioned, this was the focus of my PhD, but there's an enormous research in the field, and how changes in our immune system are eventually also affecting the way our brain functions, which makes sense, of course, that we need to adjust our behavior, and maybe the sickness behavior is the best example, to the demands of the organism. But my lab is mainly focused on the other way around, on how the brain controls the immune system. And I think maybe the most fundamental question to ask, especially in this audience, is why? Why should we even give the nervous system anything to say on the immune system? Because for years we studied the immune system and it works very well without neuroscientists getting in between. And I think the question that we need to ask is, what is the advantage? What is unique about the information that can be provided by the brain? And I will talk about three main things very briefly: integration, speed of reaction, and prediction. And what do I mean? When I'm talking about integration, it's all about we kind of break down the response to immune response, metabolic response, behavioral response, but eventually in the body, it all has to be synchronized. We have to maintain a proper temperature. You need to allocate the resources in terms of metabolism to allow an immune response. You need to adjust behavior accordingly, and also to integrate inputs from outside and inside. And all of that can be orchestrated by the brain, which is the basic role of that. So eventually the brain can synchronize between the different systems, including blood flow. Everything can be synchronized eventually also by the brain. The other thing is speed of reaction. And I think it's really important to remember that the immune system has a pace that is very, very different from the time scales of the nervous system. And it's an important pace. It has to be relatively slow, so that there are a lot of control mechanisms into the immune system that are embedded in between. But sometimes you need a very, very fast response. And this is something that you cannot achieve with the classical systems that we have in the immune system of cytokines and communication between individual cells. These are mainly localized responses. But sometimes you need something fast, something systemic. This can be in the case of stress, when you have a huge stress, and then you are in the risk of having some kind of damage. You don't know where it's going to happen. And one of the things that we see that happens in physiology is that stress mobilizes immune cells to the blood. And now they're more accessible to whatever is going to happen. So this is for the initiation of response, but sometimes you need also to shut down the response very fast, like in the case of sepsis, when the immune response is overwhelming, and the only thing you can do is to shut it down immediately all over. And this is indeed the case of inflammatory reflex that Kevin Tracy showed many years ago, showing that when vagal stimulation can suppress the immune response altogether. And this can be a mechanism when you need to shut down the system. And finally, which was the focus on our lab mainly, is the prediction, because the brain, in essence, is learning from the past, so it can initiate the most effective and fast response when something else is happening in the future, or anticipate that something is going to happen and prepare the appropriate response. And this can happen and in the case of innate responses. And we need to remember that the immune system, the immune system will respond only after it saw the challenge, the antigen. But sometimes you can anticipate that you're going to encounter a challenge. And these are kind of innate programs. It can be innate programs. So our ancestors already made the connection evolutionary between stress, for example, and the potential damage. So then stress mounts and the mobilization of immune cells to the blood. Another example, this was the focus of our initial work, was the reward system. So when you anticipate that you're going, the reward system in the brain is being activated when you have anticipation of important motivated behaviors like mating and feeding. So these are points that you are exposed to pathogens. And indeed, we see that activation of the brain reward system boosts immunity. Another very active field of research now in the lab is sensory information. For example, the information that we have that comes with the food. This is kind of information that is very relevant to the appropriate immune response that you can mount. But these are kind of informations that I think many of them are innate, meaning that they acquire throughout evolution. But sometimes we also learn in our individual lives. And this is a process that I think we acquire new information. And this is these connections that we make between one experience and another physiologically, like Pavlov's dog, right? So they made the connection between the bell ring and the fact that they're going to get food. So their gut is going to start to become activated in anticipation of the food. And then you get a more effective processing of the food. So the same thing can also happen in the immune system. And this was actually shown in the 1920s by Metalnikov and Korin. Then it was rediscovered by Ader and Cohen. And I think most of the modern work was done, a lot of it by Manfred Szydowski. So what is the concept of immune conditioning? The concept is that you can give rats, mice, humans an immunosuppressive drug, but present it every time with saccharin. Saccharin, immunosuppressive drug, saccharin, immunosuppressive drug. And then at some point, it's sufficient to give saccharin and the mice, rats will respond with immune suppression. This is not the classical memory. It's a memory, right? It's clearly because you associate between a saccharin and a specific immune response, but it's not the classical immune memory. It's almost like memories in the brain, right? And so Tamar Kuren in the lab, together with Kobe Rosenblum's lab, started thinking of this idea. And she said, maybe there are sort of memories that we acquire throughout life that will create associations between certain conditions. For example, every time I go to drink in this well, I get salmonella infection. So maybe I should anticipate and prepare an appropriate immune response. So she wanted to look for this kind of representations in the brain. So to do that, she needed to create a challenge, an inflammatory challenge. And we decided to focus on gut inflammation in a model of DSS-induced colitis. And of course, the rationale was that the gut is very active neuronal site, where nervous system is highly innervated, It's also very active in terms of immune activity. And we know that many diseases that are related to the gut, inflammatory disease, have a component, a strong emotional component. So it seemed like a good challenge. And in DSS-induced colitis, we give mice DSS in the drinking water. This causes damage to the epithelial wall. And then the ensuing inflammatory response is actually actually causing the damage. Now we wanted to see what happens in the brain. How is it represented in the brain? And for that, we use a model of trap mice. These mice allow us to capture neurons that were active at a given time point. And the way it's done is that neurons that are active, once they're active, are going to express immediate early genes. So there are different models of that. But the idea is that at the time point when they express immediate early genes, and there is tamoxifen in the system, which you control. So it's all the time you have neurons that are active, but only the neurons that are active when you injected tamoxifen, these neurons are going to express Cre. And once they express Cre, now you can drive anything you want under the Cre promoter, in the expression of Cre, which is Cre dependent. And that can be done, for example, to express fluorescent reporter. And we used trap mice that were bred to a reporter, fluorescent reporter. And now we gave, in the peak of the DSS inflammatory response, we gave them tamoxifen. So we are going to capture the neurons that were active in the peak of the inflammation. And we see, what you see here is these red dots are actually expressed fluorescent staining in neurons that were active in the peak of the inflammation. And you can see that it actually across the brain, many brain areas are involved. And but we decided to focus on the insular cortex, this area. And the reason we decided to focus on this insular cortex, because this is a very interesting site. The insula is kind of a hidden part of the cortex, and it's involved in a process called interoception, meaning the formation of an internal image of how do I feel. This is the Bud Craig called it, how do I feel? And of course, it almost doesn't make sense that the immune information will not be part of this how do I feel. And then anatomically, the insula receives inputs from peripheral neurons and sends inputs to peripheral organs, immune organs as well. And functionally, Manfred-Chudovsky showed that lesions to this area disrupt immune conditioning. So it made sense that it will be a target. And indeed, we showed that, yes, in the insula, we see more neurons active in the peak of the inflammation. So, but the fact that there are neurons active there doesn't mean much. The question is, are they meaningful for immune regulation? So to address that, we decided now, instead of expressing, so now we decided to focus on the insula. We want to manipulate the neurons that were active in the peak of the inflammation. And in order to manipulate them, because these neurons now express Cre, right? These are trap mice. So I can capture neurons that were active in the peak of the inflammation. But now, instead of just expressing a fluorescent reporter in these neurons, I can express in these neurons DREADS. DREADS is a form, or it's a receptor, it's a mutated receptor that allows to activate the neurons, that it will activate the neurons once I'm going to inject a drug. In this case, it's CNO. So now I have basically a remote control in which I can reactivate these specific neurons. So we are targeting the insula, expressing it to express the DREADS, the information for DREAD. We wait for 3 weeks for mice to recover. Then we give them DSS, we induce the inflammation. In the peak of the inflammation, we're going to give them tamoxifen, so to capture the neurons that were active there. Now these neurons are going to express the DREADS, and then we let mice 4 weeks to recover entirely. They recover, and then we give them CNO that will activate these DREADS. So I'm going to reactivate the neuronal ensembles that were active in the peak of the inflammation. And then when I'm looking on the immune system, I'm going to see what happened. And this is what happens. So what you see here, this is staining for CD45 of colons of control mice and GQ mice that we activated them with the DREADS. So you can see that just by reactivating the neuronal ensembles that were active in the peak of the inflammation, it basically generates the inflammation. And this is a comparison. So we compare it to the original inflammation in terms of different compartments of the colon and the lamina propria and the IELs. And what you can see is that actually it also, the reactivation of these neuronal ensembles also generates inflammatory response, at least in cell numbers. We also compared it to the original inflammation. So what you see here is kind of like an overview. So what you see is this is what happens in the original model, and this is once we reactivate. So you can see that things are going in the same direction, many parameters. So we check in different components, and it actually recapitulates many of the features of the original inflammatory response. Not everything. In some cases, it was not in the same direction. Some factors, or maybe at least like not to the same extent. We could also see not only in cell numbers, but we could also in markers, the cellular markers, but also in cytokines that are expressed, that it recapitulates many of the features of the original inflammation, but it's not exactly the same. Okay, so it seems to be meaningful. Yes, these neurons are actually doing something. But what was my control? My control was sham virus. Sham virus meaning I did the surgeries. I didn't do it, but Tamar did the surgeries, and they went through viral injection and they got CNO. But in their case, there was no dread expression. So these neurons, actually, it's almost like activating the insula and not activating the insula. So maybe there is some sort of innate information in the insula. So now what we did, we repeated the same paradigm, but now we had 3 groups of mice. Each of them expressed GQ. So in each of them, I'm going to reactivate the neurons. The difference is going to be at what point I'm going to capture them, either before the inflammation. So I'm capturing the neurons that are active before the inflammation, during the inflammation, or after the inflammation. And all of them, after they recover, we are reactivating the neurons. And you can see that only once we reactivate the neuronal ensembles that were active in the peak of the inflammation, we get the reactivation of the immune response, of the inflammatory response. But again, one can say, if you remember, I showed you that there are more neurons active in the peak of the inflammation. So you can say, yeah, but you're just activating more neurons. So we control for that. And now we just activated neurons without trapping. Just for comparison, this in green, you can see the number of neurons that we're activating in general, when we're using a general activation, and in red, it's when we are activating only the trapped neurons. So you can see it's about twice the number of neurons that we are activating, and nothing happens. So there is something specific in the neuronal ensembles that are activated during, in this case, that were active in the peak of the inflammation. It's not a matter of how many neurons I'm activating. In fact, one can say, and that in this case, the neuronal ensembles that were active in the peak of the inflammation should potentially be also activated here in the broad activation. So part of it may be that, and we know it from memory engrams, from the field of memory engrams, then when you activate more neurons that are not related to the specific ensemble that encodes the specific memory, then you interrupt with the reactivation of the memory. So it actually speaks more to this direction. So yes, it seems like a meaningful activation, but maybe it's something that is unique to the gut-brain axis. And to answer that, now we use the zymozyme-induced peritonitis. So this is a completely different model of inflammation. In this case, we inject mice with zymozyme, which is toll-like receptor 2 agonist. And we give them, and then it's a different site. So it's peritoneum. It's a completely different type of inflammation. And now we are going to look at the activity before and after. So we're capturing neurons before and after, and sham. So mice that didn't go any inflammation. And what you can see is only when we are reactivating the neurons that are active in the peak of the inflammation, we are getting the inflammatory response. So it's not specific to the gut-brain axis. I can tell you also that if we look in the colon, we don't see any effect when we're reactivating the neurons in the that were active during peritonitis. So it has some specificity, anatomical specificity at least. And then there's the question of how how are these effects actually mediated? So I'm capturing these neurons in the insula, and I'm getting these very specific reactions in the colon. How is it happening? So in this case, we got, with the help of Oren Kobiner, we used PRV virus. So this virus goes retrogradely. Basically, I can inject it in the colon, and then it will jump several synapses all the way to the brain. And then I can see the trace in the brain. And indeed, you can see the trace in the brain. You can see it in the insular cortex. You can see also that in the amygdala, in the hypothalamus, which are all areas that were actually connected to neuronal, to brain-related control of immunity. And what it means is that these neurons have some anatomical connection. It's not a direct connection, so it might be many synapses in between. But remember that we understand that there's something unique about the neuronal ensembles that are active at a given time point. So is it just random neurons that were activated in the insula? So now what we did was trapping. So we captured the neurons that were active in the peak of the inflammation, and to the same mice, we injected PRV. And then what we can see is that there is quite significant overlap. So neurons that send projections to the colon are also neurons that were active in the peak of the inflammation. Not all of them, of course. And then we did the same thing for the peritoneum. And there in the peritoneum, it was a problem, and we could not get this. So we had some limitations when we were working with the peritoneum, and we decided to shorten the duration, so to kind of bridge the gap. So what we did, we injected to the insula, in the trapped neurons, we injected a virus called AV1, which will jump one synapse forward. And then we injected the second virus to the peritoneum. And now we want to see where they meet. So what is the kind of the pathway that is going from the insula to the peritoneum? And what you can see is that they meet in two very important, they meet in several points, but two very interesting are DMV and RVLM. The DMV controls the vagus. The RVLM controls the sympathetic nervous system. What does it mean? It means that there is anatomical connectivity. So the neurons that are active in the peak of the inflammation are neurons that also project to the DMV and RVLM and can control these two peripheral autonomic nervous system. It doesn't mean that it happens functionally. It's just an anatomical connection, and we still need to prove what happens in terms of function. Is it a balance between the activity of these two systems? What is happening there? So it seems that it's mediated via the autonomic nervous system, but I think maybe the big question is, how do you get it so specific? How do you get such a precise response either in the colon or in the peritoneum? So Maya Schiller and the lab started playing with this idea regarding the autonomic nervous system. So the autonomic nervous system, especially the sympathetic nervous system, we often correlate it with stress response. And we already know that many of the effects that we see in our brain manipulations, especially activation of the immune system, are mediated by the sympathetic nervous system. The thing is that the sympathetic nervous system, the way we usually look on neuroadrenaline in the blood, but this is a systemic response that is mediated by the adrenal gland. But there are also local sympathetic innervations to all immune organs. So Maya said, well, maybe there's something unique. Maybe the information that is delivered by the sympathetic nerves that go to the specific sites, maybe they have some unique information and something different in terms of controlling the immune system. So she developed a model in which she crossed mice that are channelrhodopsin, to express channelrhodopsin, and in a Cree-dependent manner. And channelrhodopsin allows us to, basically it's the optogenetic channel, and the Cree was under tyrosine hydroxylase. Tyrosine hydroxylase is the one required for sympathetic neurons for the synthesis of neuroadrenaline, but also other factors, catecholinergic, other transmitters. So it's actually going to be expressed in all catecholaminergic neurons. We control for that. So I'm going to talk only on the effects of the sympathetic nervous system. But now, if I'm going to shine light on these neurons, and this is what you can see that this, for example, this sympathetic ganglions express channel rhodopsin, I can activate them. We wanted to activate the neurons in the colon. And to do that, we insert, Maya had this idea that we can insert an anal probe of the optogenetic channel, and then we will stimulate only the neurons in the colon. And you can see this is analysis of neuroadrenaline levels, and you can see that post-stimulation, they increase the number of neuroadrenaline, but it's very local. It's not overall, and it's not in the serum, for example. It doesn't happen in littermates first. So it's specific, and by shining this light, she can activate these neurons. And then she gave them DSS-induced colitis, and every day she also used optogenetic activation. And you can see that the optogenetic activation actually had beneficial effects on the recovery in the model of DSS-induced colitis. So you can see that in controls that just get DSS, there is a more shortening of the colon. But in those that were also received DSS, but were also treated optogenetically, had reduced levels of inflammation. And the question is, how does it make sense? Because we know that stress usually exacerbates the inflammatory response. But it's important to know that the reports are really kind of mixed in this field. So what she did, and just to show you that there is indeed reduction in the immune cells in this site, and we tried to identify which cells it happens with, but we couldn't. So just to show you that it's just an overall kind of effect, until we realized that the effect was actually happening on the channel, on the blood vessels. Because what you see here, CD31 is the staining for blood vessels, and the channel rhodopsin is the optogenetic channel. And what you can see is that it's expressed around blood vessels. And so what Maya started to understand what happens there, and she found is that activation. So one of the things that we know is that when immune cells are in the blood, in order to extravagate to the tissue, to generate the inflammation locally, they need to go the process through several adhesion and processes and crawling and then extravasation. And it's dependent on certain adhesion molecules. And what she found is that activation, optogenetic activation of these fibers, sympathetic fibers, reduce some of these molecules, specifically MATCAM1 and ICAM1, which are required for this extravasation. We could also see the same phenomena if we just treat endothelial cells with neuroadrenaline. The interesting part is that it has to be very high concentration of neuroadrenaline. And I think this speaks to the difference between systemic neuroadrenaline and local neuroadrenaline, because systemically you cannot reach these high levels of neuroadrenaline, but locally you can secrete quite high, which is actually, and then the effect remains there. So you use the same mediator, the same neurotransmitter to generate two very different reactions. And then if we're talking about mobilization and local signals extravagate here, then it's something that you can generate very local response and very high levels, very local. So this is just one example how local innovations can do that. And then the question is, okay, so what happens in the brain? It's one possibility. Indeed, we understand that it's mediated, at least in part, by the autonomic nervous system, but what happens in the brain? For example, are they the same neurons that are being activated every time that I'm going to give mice DSS-induced colitis? Am I going to get the same kind of response? And what you can see here, this is we trapped neurons initially with trapping, and then we gave them again DSS, and now we analyze CFOS. It has some technical limitations, but you can see that even just by first analysis, it doesn't seem that it's the same neurons. It's in the same area, but it's not the same neurons. Another interesting thing that we are seeing now, and this is very preliminary, but we see that if we give several inflammatory reactions, even the nature of the information and the different brain areas that are being activated upon repeated exposures are very different. So it doesn't seem that it's the same neurons, which is actually as it happens with memory engrams. Another type of information that may be relevant and it's encoded in the brain may be related to pain. So we know that inflammatory reactions are accompanied by pain, and there is almost no model of pain in which you induce pain and there is no inflammatory response that follows. So what we did, we now captured neurons that were active in the peak of the inflammation, but in the presence of analgesia. And when we reactivated them, we could see that it didn't recapitulate exactly the same effects. So there is at least some information that is encoded by these neurons. So we know that in the brain, the information may be within specific neurons, and it's at least in part also dependent on pain, which is a very interesting question on its own. Another thing that we are very actively exploring now is to understand, for example, what are the connections from which brain areas these specific neurons that are active during inflammation, which areas talk to them and where these neurons send connection, and what is unique in the case of inflammation. So does it change, or is it like a general flow of information from the same site to the same targets? And finally, if in general, activation of the insula is something that happens in the brain during inflammation, what is the relative contribution of that to the ongoing inflammatory response? And maybe there is a clinical potential there. So what we did now we injected to the insula DREADs, but the inhibitory form of DREAD, the one that when we inject CNO, now it's going to inhibit the activity of these neurons. And then we gave them DSS-induced colitis, so they actually have damage, but only we're blocking the information that is encoded in the insula. And what we see now is that we can attenuate to very limited extent, I think, the clinical symptoms, but in terms of the number of immune cells that infiltrate, we see less with this in this case. So what does it mean? One of the things that we see is that there is immune-related information that's stored in the brain, almost in mnemonic-like representation. And this maybe makes us think whether there is some sort of different form of immune memory, which is not limited to antibodies or immune cells, and maybe also partially represented in the brain. And when I'm saying immune-related information, it can mean many things. For example, it can mean that I don't know if what the brain records is the state of the tissue or the immune information, because I'm only following the effect on the immune system, but it might be that the information that is stored in the brain relates to the tissue itself. But it has, I think, functionally, what is really interesting is that it has a lot of implications for psychosomatic disorders, because we know that there are many cases in which patients have an inflammatory response that is triggered by an emotional mechanism. And I think one of the most interesting examples is a clinician in the 1800s. They described his patient that she is allergic to pollen. But even when he presented her an artificial flower, she responded with allergy. And in fact, we can see cases of patients, for example, with Crohn's, when they come and they can say to the clinician, oh, I had like an emotional episode. I know now I have a flare. If you check them clinically, there is no flare. The flare comes about a week later. So the feeling of the flare actually precedes the actual flare. So, and maybe if we interfere exactly in this point when it begins, then we can stop the vicious cycle, because we already understand that the brain contributes to the process of the disease. And there is a question of how we generate the specificity. And what do I mean? We see that the brain, that the activation of these neuronal ensembles in the brain generate very precise reactions in terms of anatomy, but also an immune response. But here's a gap. We know that the immune response is very complex, right? Many factors, many parameters. The nervous system has enough of the complexity to generate this, to encode this complex immune response. But then once it sends the information back to the periphery, it's probably why this sympathetic and parasympathetic nervous system, which have quite limited capacity. So the question is, how is this complex information delivered to the tissue and generates again all this complexity? So what we suggest is that actually some part of the memory is stored by the tissue itself. And this is something that we are looking into now, because it might be that after tissue undergoes some inflammatory reaction, it changes a lot of its properties. For example, changes in the expression of neuronal mediators, receptors for neuronal mediators, the neuronal innervation in the tissue, and the cells that remain, the immune cells that remain there, and can trigger reactions differently after the tissue already encountered, and actually be tuned differently to the dialogue with the nervous system. And I hope that next time I can show you more about that. So what we are arguing is that in contrast to the engram, the memory engram that we know, as we know when we encode the memory, the immune engram is something that is distributed. No, it's not limited to neurons. It's actually distributed between the brain and the tissue. And I think finally, if I'm taking it back and to close the whole story back to what I started with, this idea of immunoception, that there is an ongoing process of of programs that are both innate and adaptive. And the innate programs is something that we actually acquire through evolution, but the adaptive one is that are unique to each one of us. And they are, in order for them to be executed, there is an ongoing process of immunoception in which the brain monitors what is the state of the tissue or the state of the immune response, and then can send back corrective messages that will be synchronized with the entire organism. So with that, I would like to thank all the people that were involved in this work, our collaborators, and our funding. And I will be happy to answer the Twitter questions. And thank you for listening.
Thank you so much for such a wonderful talk. And just noting the, would you like to join us on your closing slide? Yes, I would. And I'm sure that so many people would too. So I'm sure everybody's got so many questions for you, and we won't be thinking of immune memory the same way again. Anyone who would like to ask Asya a question, please go to our Q&A on Twitter, search for the account Global IMMUNOTALKS, find the tweet that says ask questions for Dr. Asya Rolls here, and reply to that tweet with your question and mention the hashtag Global Immuno. And Asya will be responding for a little while after this talk, using the Immuno speaker response. So thank you so much for such a wonderful talk. Just to highlight to everybody, next week we'll have Shimon Sakaguchi joining us, so please tune in again for that. And thank you so much again to our wonderful Immuno speaker, and we'll look forward to seeing everybody next time. Thank you.